Okay, good afternoon. Thank you everybody for joining us today. My name is Stuart Newman. I am the CEO of Therapeutic Innovation Australia, and it's my pleasure to welcome you to this webinar, Accelerate Your Translational Research with TIA. Just want to commence with acknowledgement of country, and I'd like to acknowledge and pay my respects to the Yagara people, the custodians of the land on which many of us, or not all of us, gather today, and pay my respects to the elders past, present, and emerging. Just before we kick off, a little housekeeping. We are delighted to answer your questions. Please ask them via the Zoom's Q&A function and not the chat window. And we'll address questions either in real time or we can address them at more length after all the speakers have spoken. And after the end of this webinar, a short, and I really mean short, survey will be sent out to you with a few follow-up questions to get a bit of a feel for the demographics and where people are dialing in from. And lastly, this webinar is being recorded and it will be hosted on the TIA website in a week or so. So it can be sent to any of your colleagues who are not able to join us today. So I just want to give a bit of a rundown of the structure of this webinar. First, I'm going to be introducing TIA and NCRIS at a high level on what the aims of the organization are. Then each of our three TIA capability leads will describe each of the services available within each capability and how they can assist your therapeutic development research. Then I'll come back to myself to talk about how to access these capabilities, either through direct approach to the facilities or our staff, and also how to enable access via our Pipeline Accelerator scheme, which we launched 2 weeks ago, which closes on the end of October. And finally, I'm delighted to have some testimonials, some satisfied customers, some users of the Accelerator scheme, Professor Alex Loukas of James Cook University, and Mr. Greg Santamaria of Cyclotek Proprietary Limited, a university and SME-based users of the voucher scheme. So without further ado, I'd like to just start off by giving a bit of a big picture on what is NCRIS. Now, NCRIS is a bit of a mouthful. It's the National Collaborative Research Infrastructure Strategy. It's a Federal Department of Education program that invests in a network of research infrastructure projects with the overall aim to provide access to national research infrastructure to researchers across the country, whether they are from the public or private sector. It's received a lot of long-term bipartisan funding over a long period of time, and there are NCRIS research infrastructure facilities across all the areas of science in which Australia is or seeks to be world-class. And the final point there in bright red friendly letters, the C in NCRIS is collaborative, and it's the heart of everything we do. So on that score, there are a number of people like us in the NCRIS network. And I'd just like to very briefly introduce my NCRIS cousins. The National Imaging Facility provides a very wide range of biomedical imaging, MRI and CT, and life science space. Population Health Research Network provides access to linked data across a number of jurisdictions to aid in epidemiological, public health, and clinical trial design. Bioplatforms Australia is omics of all kinds, proteomics, metabolomics, and genomics, as well as bioinformatics, horsepower to drive and inform discovery and translational research. And finally, Phenomics Australia are a source of commonly used and novel production of mouse models of human disease, and they're also expanding into iPSC cells and other models of disease for research and development purposes. So any of these projects, if they're of any interest to you, get in touch with us and we can put you in touch with the right people in the right place. But back to us. Therapeutic Innovation Australia, we as an entity are a not-for-profit company, so we're independent. We're established primarily to manage government investment from the NCRIS program in research infrastructure. We've been around a little while now. It's only for the last maybe 2.5 years where we've had the structure and focus that we have at the moment. So where do we put ourselves? Where have we plonked ourselves in the therapeutic development pathway? As you'd all be aware, development of therapeutics is long and risky, probably going to fail, it's very expensive, but if successful can have massive social, economic and health benefits to Australia and indeed the world. But there is this valley of death that is persistent and is where a lot of projects with high potential fall down either through lack of funding, lack of support, lack of available capability, or lack of knowledge as to what to do next. So TIA has put ourselves right in the middle of this valley of death to do what we can to help projects over this valley and to reach the sunlit uplands past this IP inflection point and into phase 1 and potentially phase 2 clinical trials. And we do this by managing large-scale infrastructure funding at a variety of specially chosen research infrastructure facilities across the country, and we'll go through who they are and what they are a little bit later on with the aid of our leads. But in general, we invest in people, key salaries of technical and scientific support staff at facilities. We also invest in equipment at these facilities to enable the staff to do what they do very well. And we also make some investment in the voucher scheme to enable additional access, which I'll talk about a little bit later on. So how do we organize ourselves? We essentially, at about 2.5 years ago, reorganized ourselves into 3 product development pipelines focused on 3 different therapeutic modalities. We did this because each of these modalities are either mature and have a strong track record of success or are emerging and have great potential. We chose small molecule pharmaceuticals, biologics and vaccines, and cell and gene therapy space. Now, with these, under these 3 groups, we have around 24, maybe 25 by now, national research infrastructure facilities that support development in these 3 particular modalities. As I noted earlier, access is for researchers, whether they are from SMEs or universities, or CSIRO, or medical research institutes, or from overseas institutions as well. And the key thing is that each of these capabilities is supported by a project chaperone, or a capability lead, as we call them, and you'll be meeting each of our 3 capability leads during the course of this webinar. So just before we get to that, I just wanted to make the point that the access that TIA invests and enables is in the experience of the people and their expertise, and also the equipment to support them. 91 staff across the 25 or so facilities. Where appropriate, many of these facilities have quality accreditation to make sure that the results that they produce are reliable and give a lot of confidence to enable further investment. But I really don't want to get to our capability leads. So first of all, I'd like to just introduce, actually no, what's going to happen, I'll in turn, our 3 capability leads will spend a bit of time talking about the services available and accessible in their particular capabilities. And we'll start off with small molecules with John Paraso, followed by cell and gene therapies with Heather Donaghy, and finally biologics and vaccines with Ben Hughes. Just so you have a bit of a sense of where they are. John is in Melbourne, Heather is in Sydney, and Ben is in Brisbane. So we really are scattered all up and down the East Coast at the moment. So if I could just ask John to share his screen, I will stop sharing mine, and tell us a little bit about the small molecule capability of TIA. Thanks, John.
Thank you. All right, sorry for that little hiccup. I think I'll need another 5 years of COVID before I get this completely mastered. So today, thank you, introducing the Therapeutic Innovation Australia's small molecules capabilities. I'll go through some slides here just to describe what the network entails. So on the next slide, Stuart. Just wanted to highlight here the significant areas of development within the small molecule drug discovery field. As I'm sure many of you know, when projects come out of academia, medical research institutes, and so forth, they may be on the lookout for either tool compounds or therapeutics. And in both instances, this is the pipeline, that it will, and the pathway that it will follow. You would consider screening for compounds that may, small molecules that may inhibit your target of interest, in which case you'll need libraries to be able, of compounds to be able to screen, and need people to help you with this compound storages and logistics and dispensing volumes down to even nanoliters in many instances. You'll need to talk to the high throughput screening facilities that can handle your type of screen, whether it's a target-based, cell-based, phenotypic or fragments libraries that you're looking at. You'll also eventually hopefully have some hits that you'll need guidance from medicinal chemistry people at which I'll describe shortly, that have the background into how to make your hit much more amenable to being a drug-like compound, because it's very rare that well, almost impossible that you'll have a hit which turns into a drug without much optimization. As you can see through this pipeline, from medicinal chemistry, there's constant interaction with running the compounds in drug metabolism and PK/PD models, either in vitro and ultimately in vivo as well, with animal efficacy models, looking at biomarkers that could be a readout of the effect of your molecule, and ultimately right up to preclinical, putting a package together to convince the FDA to allow you to move your compound into the clinic, including GLP, safety, tox, active pharmaceutical ingredient formulation, and stability. So all these areas follow a pipeline, a pathway, which is quite complex within small molecule drug discovery and development. So the next, Stuart. So what the TIA have done over the years is build up a network of small molecule capabilities entailing many facilities. So we've got, early on in the pipeline, as I mentioned, you need access to compounds for screening. So we have within direct support for Compounds Australia, who look after our libraries, which I'll go to in a little bit of detail later, and the logistics of handling all the compounds. We also have voucher supported HDS network, which I'll describe, and Stuart will be describing the voucher scheme in more detail later. So from the screening teams, as I mentioned, there's a strong medicinal chemistry component, so it's covered by the Australian Translational Medicinal Chemistry Facility at Monash Parkville. Also, they're collaborating with the Centre for Drug candidate optimization, could do a lot of the drug metabolism and pharmacokinetic guided lead optimization in that phase. And prior to getting to an IND package, you would need to speak to the Center of Integrated Preclinical Drug Development to help show that your molecule works in vivo efficacy models of whichever relevant therapeutic area, and also that your data is obtained in a quality management system. Lastly, we used to have also TetraQ up at UQ that would help with us do preclinical GLP toxicology, but they have recently, well, 6 months ago, been sold, which is a great story. They were sold to Agilex Biolabs. And so in the future iterations of the small molecule capability network, we'll be looking to help fill this area further on. The next slide, Stuart. Yep, thank you. So this is just to give you an overview of the whole national network, including the core facilities that I mentioned, but also in this particular slide, the high throughput screening network that we work closely with in the voucher scheme, but also in many other projects that come through with TIA support on one form or other. So up in Queensland, you see we have the Compounds Australia team that handle the library storage and stamping out of the libraries. The Centre for Integrated Pharmacology of Disease at UQ, as I mentioned before, that run our efficacy models. Across down to Melbourne, there's the Australian Translational and Medicinal Chemistry Facility and the Centre for Drug Candidate Optimization at Monash Uni. And within each of these rectangles, I've also described the high throughput screening networks that we use and interact with within our pipeline. So there's Griffith University Institute for Drug Discovery up in Queensland, Brisbane. And at UQ, there's also the Community for Open Antimicrobial Drug Discovery. Further south in Sydney, we have the Children's Cancer Institute, CCIA. We also have the Victor Chang Cardiac Research Institutions Innovation Centre with a high throughput centre there as well. In Canberra, we have the ANU Centre for Therapeutic Drug Discovery. And across to Adelaide, the Cell Screen South Australia at Flinders University. And last but not least, in Melbourne, we're all familiar with WEHI, with the National Drug Discovery Centre, and PDMAC, which has been involved for many years with the Victorian Centre for Functional Genomics. And in the past few years, of course, there's also a strong emphasis at Monash, at Parkville, on looking at the Monash fragment platforms. So altogether, these networks, and we're missing one in Perth, and hopefully we'll get one in the future, will provide the advice and also the work to be done to get your compound through to a therapeutic. So next slide, Stuart. It's just a quick summary of our core facilities here. So just briefly on the Compounds Australia network, they have a collection, some open, some closed, but can be accessed for memberships of compounds. The open scaffold collection of 33,000 compounds, as you can see, an open drug collection, which contains a lot of FDA compounds, an academic collection, which is predominantly comes from academic research across Australia, CSIRO included. Also a strong focus on Nature Bank, which is compounds that have come from either extracts or compounds that have come from the screening up in Griffith. And lastly, there's an Australian Lead Identification Consortium, which is the largest library in Australia of over 200,000, representing a quite diverse lead-like series of compounds to screen. The next slide, Stuart. So this slide is just to show you the interaction that happens between the Australian Translational Medicinal Chemistry Facility and the Centre for Drug Candidate Optimization at Monash. So when you have some hits from your compound, there's a lot of work to get involved in chemistry to get them into higher potency, better selectivity, and also better properties in terms of drug metabolism. So there's a lot of interplay between these two centres, and having them both at Monash at Parkville helps enormously in progressing your project through in a quicker timeline. On the next slide, Stuart. And last but not least, so their core facilities is CIPDD, as I mentioned earlier, that are strongly involved in vivo efficacy assessment. They are a NATO accredited facility that can test your compounds in accredited pain and some diabetes models as well, and also recently multiple sclerosis is ones that we're adding to the pipeline. So these animal models are important because without them, you do not have the efficacy data in a quality system to be able to put your IND package together to get your compound through to the clinic. So they're the core facilities and a quick overview of all the HES facilities in our network. I think on the final slide, I've just got my contact details, and I encourage everyone in the small molecule capabilities area, no matter which part of the pipeline you wish to access or discuss, I can put you in contact with the right people. Apart from the knowledge I have, there's the network behind me in all these areas, and the people that I know here within TIA and within this network can help you progress your project very efficiently. Thank you, everyone.
Thanks very much for that, John. I just want to note before we move on to our next speaker that John is shortly going to have some space at the Bio21 Institute in the heart of the Parkville precinct, where TIA is moving its Melbourne office from the CBD Collins Street to Bio21. So if you're in that area, have a look out for some TIA logos sprouting up into the foyer, and pop in and say hi. And so thanks very much for that. Next speaker, I'd like to invite Heather Donaghy, who's based in Sydney, to talk a bit about the cell and gene therapy capability. And Heather, hopefully you'll be able to share a screen. Fantastic. Okay, take it away.
There we go. Right, so I'm going to talk about the cell and gene therapy capability within TIA. These products are kind of a different scale to the small molecules that we're talking about. They're a lot bigger in their size, but they're also, excuse me, a lot smaller in their manufacturing processes. So rather than scaling up to make a larger product, a larger batch of your product, you would want to be scaling out to be able to make lots of batches, because each batch tends to coincide with one patient. However, they are, like the small molecules, they're also very technically difficult to make, and even more so because you're doing it on that individual basis. So on the left-hand side here, I have a kind of a flow diagram of what would happen with a cell therapy, for example, maybe a CAR T, where you take the blood from the patient, you isolate specific cells, you then transduce those cells with the specific vector to express the CAR T that you would like them to express, expand out those CAR T expressing cells, harvest those cells, and then infuse back into a patient. Conversely, if you're looking at a gene therapy down on the bottom right hand side, then you would start with a cell culture. Then you will add your virus containing your vector into your cell culture. You'll expand out that that vector production, and then purify out your cells, purify out your vector, which can then either go into the CAR T in vitro or ex vivo, go into a CAR T, or an in vivo gene manipulation or gene modification and go straight into the patient. So because of all these multiple steps in each of these steps in the process of making your product for a patient, the more efficient you can make it, the quicker you can make your turnaround time, the higher the efficiency you'll get from your products. Also, because they're all derived from a cell culture environment, it's a really highly complex regulatory environment involved, from the materials that you need to do your isolation, materials that you need to do your cell culture, and ensuring that all of those are GMP compliant or GMP grade, as well as the quality control to make sure that your particular drug falls within specific parameters for perhaps you need a particular percentage of T cells or expressing a particular vector, or a number of cells that you can put back into the patient, or maybe the number of viral vector genome copies that you need to put into the patient as well. So all of these things are highly complex and for the quality control. And then of course, because you've got the in vitro culture conditions, then there's infection control, both from a contamination of your cell culture, but also from your starting material, if you're getting material from patients or from individuals and putting back into perhaps a different individual, then the infection control from that as well. So at TIA, we have a national network of cell and gene therapies focused facilities. Over in WA, there's Cell and Tissue Therapies WA, CTT WA. In Sydney, there's the CMT at RPA, and also Sydney Cell and Gene Therapies, manufacturing different therapies. QGen in Brisbane, and then Peter Mac Centre of Excellence in Melbourne. And on top of that, for the voucher scheme that we're launching, we've also introduced the vector and genome engineering facility also at Westmeads that will help you to manufacture your viral vector for your gene or cell therapies. So together, these centres offer the full range of manufacturing capability across the landscape. And these include pretty much the whole spectrum of cell, gene and tissue therapies that are currently being investigated worldwide, but we have that expertise here in Australia. So stem cells and mesenchymal cells, autologous and allogeneic CAR Ts within the cell therapy space, and then the gene therapies as well. So the in vivo gene therapies, both for pediatrics and for adults. And then also the tissue engineering and scaffolding to be able to create implantable devices and implantable products into patients. So all of the different facilities will help you with your process development to make your manufacturing process robust and reproducible. And a lot of this consists of you transitioning to a closed system rather than having a manual pipetting in the hood. You'll move into a closed system with minimum open handling to reduce the chances of contamination, but also transferring over from research grade only use into clinical grade for your media and your supplements, and moving away from animal products. So moving away from fetal calf serum, for example, and into the human serum. And then the viral vector manufacture, we have the ability to manufacture adeno, lenti, and AAV And the VGEF facility, they will also help you to identify which is the best AAV serotype for your needs. Then moving into the GMP manufacturing, when you're ready to go to phase 1 clinical trial and beyond, the facilities can help you with the manufacturing to the standard for patient treatment. And then quality control to ensure that you have that consistency and safety of your products, and make sure that you track your product throughout the manufacturing process. So each of the facilities is able to help it. It has its own specific focus, and so I would say that probably the best place to start your journey, if you're thinking of progressing your cell and gene therapy, is to speak to me, reach out to me, and I will be able to help you with identifying which of the different facilities would be the best place for you to turn for your product development. Thank you.
Thank you very much for that, Heather. Can I just remind everybody we have the Q&A open if you'd like to ask some questions along the way, and either we'll try and answer them in real time or answer them at the end. Any kind of question on anything you've just heard or anything that's presented along the way. So on that score, I'd just like to pass on to Ben Hughes to tell us a bit about the National Biologics Facility. So take it away, Ben.
Excellent. Is that sharing?
Yes.
Okay,
it's not in presentation yet.
Why is... There we go.
There you go.
Yes.
The one. Fantastic. Okay, great. No, thanks, Stuart. And thanks everyone for joining in. My name is Ben Hughes. I am based here in Queensland. I'm going to be talking about the biologics and vaccine capability. So I am based in Queensland, but I represent and try to work closely with all three of very connected and complementary nodes within Australia. So this is a national capability. As Stuart mentioned, TIA has been around since 2013, but NCRIS funding has actually been flowing into some of these nodes since 2006. So it's really many years and a great sort of legacy of research infrastructure and delivering solutions for biologics front. So the three nodes are AIBN at the University of Queensland, the Biologics Innovation Facility at UTS in Sydney, and the Cyro Manufacturing Group down in now Clayton. I used to be in both Parkville and Clayton, but now focusing everything into the Clayton campus. So yes, similar to how John and Heather mentioned, there's a lot of complexity. And I think the old example of, maybe a small molecule is, akin to a bike, and a large biologic like a monoclonal antibody might be more like a commercial airliner. There's a lot of moving parts. There's a lot of complexity, a lot of things that can go wrong if you're not careful in how you sort of design things. And so really when it comes and the steps that we like to take when working with researchers and small to medium enterprises is really at that front end stage, try to think about the great research that's happening, but look at the end goal of, how does this, hopefully get to a therapeutic endpoint. And while you might be starting with a small volume, the decisions you make now in moving it from those, 10 to 20 mils into, a 50-liter bioreactor, sort of pictured here, and ultimately, if things are successful, into 20,000-liter bioreactors, you really have to start with the end in mind. You need to understand, what are the critical parameters, what's going to make this happen reproducibly each time. As Heather was saying, the right choice of raw materials, steering away from certain things that work absolutely fine in a lab setting, but when you need to move this into a clinical setting, you have to be aware of certain requirements. So the sort of just you know, the facilities do, as I sort of said, work very well. And there are quite a few programs that are moving through, which might actually start at one of the nodes and then move to another node, you know, from a discovery phase to production phase, and doing that under an appropriate quality system. But, you know, suffice to say, each of the nodes has sort of specialities. I'll sort of talk to some of those on the next slide. But really, you know, if you have a protein or an nucleic acid that needs to be manufactured, please feel free to reach out to myself or have a look at the website. And we have a lot of different tools and platforms and abilities to try and take your research to that next level. Maybe it's not going to go all the way to the clinic, but it's about getting to an endpoint where you can get to the next level of funding and you can start to, you know, understand, as Stuart was saying, you know, is this going to make it through the valley of death? Is this, you know, does this have value to continue? And so one thing that is new and kind of exciting since we last had a webinar is really that we have been very lucky to bring online some of that strategic funding and to bring forth some ability for Australian researchers to access nucleic acid manufacture. So this is really an investment from both TIA and the University of Queensland. It really leverages the National Biologics Facility sort of infrastructure and sort of know-how, and really partnering that with the protein expression facility also here at the Australian Institute for Bioengineering and Nanotechnology. And PEF has some you know, great work streams and has been around for a similar amount of time at working in this space. And they have some you know, excellent platforms that we're already moving into, you know, this area of nucleic acid. And so this was seen as a great opportunity to leverage this into something that we can make part of this sort of national capability. So this is sort of brought online, and we're already seeing the first customer moving through this. So it's quite exciting. So, you know, I think the summary for NBF is we do a lot, you know, and it's really about taking programs from that early, you know, research starting point, and seeing how we can add value and help with that sort of progression to hopefully, you know, a therapeutic endpoint. So you know, working with, you know, the great staff, the knowledge and the expertise and equipment at each of these facilities, you know, we can offer a value about how we design or how we identify or isolate antibodies up front, you know, how we do that design and discovery, you know, working with bioconjugation. This is another strong point that the facility has a lot of expertise and can tap into. Nucleic acid, that I just mentioned, is a new point, so it's not just about protein expression here. And while there's a lot of expertise and a lot of therapeutic proteins that are produced in mammalian cell culture, there's also a great amount of expertise within the National Biologics Facility and associated facilities around other host cells, so insect cells, microbial, yeast. So again, we have that ability to tap into a lot of different host systems and years of expertise about how we grow these at small scale and then move them to larger scale, so we can make sure that we have that scale up in mind. We have done and do a lot of process development and thinking about how to you know, logically scale these up and do this in a quality manner. A critical part for this is about how we do the analytics or the characterization. A lot of researchers are very proud of the gel that they've isolated a single band, but that gel sometimes doesn't tell the entire story of, you know, other smaller, lower levels of contaminating proteins or host cell proteins. And also just how functional is the protein that's captured there, you know. So trying to tease further into the glycosylation or how that functions in assays to show that it's actually efficacious. That's a critical part of what we do and how we can give advice and put together an appropriate testing and characterization strategy so that when we release this material for either animal studies or human studies, we're doing that appropriately so that regulators will be able to make an assessment that this is appropriate for human clinical endpoints. What we do have that's exciting is in the Cyro Clayton facility. So they've been working earnestly and bringing online a new good manufacturing practice compliant manufacturing facility. This will be up to 200 litre scale for drug substance production through to drug product, final fill of biologics and vaccines and the like. So this is will complete at the end of this year and then look for TGA certification early next year. And another point here is, yes, we also offer this sort of appropriate GMP training, and that's through the UTS facility, tapping into the NIBRT sort of internationally recognized training that's really great in this area, and how we look to upskill across the board access to this sort of relevant training. Some success stories, and there's been many over the years, but just we worked closely with Opthea many years ago now, when they were still trying to move their product through to phase 1. But now in 2019, they're one of the most valuable biotech companies, and just this year received FDA approval to move into phase 3 and continuing sort of process validation of their process as they move towards a commercial endpoint. Kira was a monoclonal antibody that was isolated and developed and manufactured, and that was then leveraged by them in 2019 into $20 million of funding to progress to a clinical sort of trial and evaluation. And only last year, both two of the 3 nodes were intensely involved in the rapid sort of showcase of how we can make a protein-based vaccine, and we were able to do that in a really short time, going from what was a DNA sequence to something that went into humans clinical trials in only 5 months, and that was leveraging extensively the NBF and TIA infrastructure capability. So this is what we can hopefully be providing to you and a lot of the other people that we have worked with over the years. So I am available. Here's my details, so please reach out, or you can use the contact us webpage for the TIA webpage. And also just on that, if you go to the TIA website right now, in the top banner that sort of rotates through different news stories, we do have a survey open. We're really looking at trying to tap into sovereign biologics capability, and the same way that we've funded more recently into mRNA capability. What are the other gaps? And so this is a survey that's open to public, and specifically anyone who's on this call might be interested in this survey. But yeah, we're really looking to try and get targeted feedback from on what are the capabilities and what are the sort of capacity gaps that we have, so that we can try and bring that into line with our forward planning. So thank you for that. I'll pass it back to you, Stuart.
Oh, thanks very much for that, Ben. That's terrific. Just on that biologics survey, what we'll do, we'll post the URL for that survey, which is on our website, into the chat window. So you can find out a little bit more about that survey as we go along. Just before I move on to talk a little bit about how to access some of these capabilities, I'd just like to acknowledge our 3 capability leads, Heather, John, and Ben. Time constraints mean I couldn't fulsomely introduce any of them, unfortunately, but we are really privileged that we have individuals of their experience and caliber, and a real diversity of lived experience in the cell and gene therapy space, small molecules and biologics. So it's terrific to have them on board. And this is the first year where we've had all three of our project chaperones shepherding projects to the right facilities. So I just wanted to point that out. I'm just going to, what am I going to do? I'm going to present a few more slides on how to access the facilities, and specifically on the voucher scheme, and then move on to our guest speakers, who I'm really keen to hear about how they fared with the scheme itself. So moving on from these capabilities, we've mentioned how to find these capabilities. What's part of TIA's very function is to put you in contact with the right facility that can help you specific to your project. And you can contact us in any way that you see fit, either through our info email address, or which is probably a bit more useful to you, is our contact form, where you can specify your area of interest, and this will funnel the inquiry to the right capability lead, and they can reach back out to you and make contact and start the conversation. You can also contact me directly, although these days I do get a lot of spam calls from the foolishness of putting my phone number on the internet, but them's the breaks. And you can also directly contact our capability leads if you have a mind to, or have their contact details already. We don't mind how you contact us, as long as you get in contact. Another way to find facilities is we have our Pipeline Navigator, which is a little searchable web app that contains details of not just our facilities, but other NCRIS and other research infrastructure facilities. You can do a bit of a keyword search on what kind of therapeutic you're interested in developing, search on the facility name, and that will give you some contacts to go and establish yourself, and a bit of a rundown of each of the facilities. I won't go through it right now because I'm keen to get to our guest speakers. So how to access TIA facilities? Well, the diversity of the facilities that we have in our consortium means there are a diversity of access models, and how one engages the services, whether it's by fee for service or a membership fee in some small cases. But in general, there are tiered pricing models, which is in accordance with NCRIS, general NCRIS principles, in that research users NCRIS funding of salaries does allow that to be subsidized. Whereas in some cases for tier 2, SME users may be charged slightly more, and full commercial costs are charged to pharma companies. But in general, advise you to talk to the facility of interest to get the lowdown on exactly how their costing model operates, because it's very bespoke depending on the project. Now, the Pipeline Accelerator is what I should imagine many of you are keen to hear about. This is a scheme that we've been running for a couple of years now, which is really designed to, as it says, get projects over the starting line. Not necessarily to see them through to completion, because we realize that projects is a very long and winding road, and we are keen to see projects go from, not necessarily from A to Z, but potentially from D to E or D to F. We have about 26 facilities that are eligible for access through the scheme, and we provide vouchers of up to $50,000 as an additional subsidy for projects. The scheme is open, as I've noticed, equally to SMEs, academics, and even some international institutions. We've had a bit of that in the past. We run these schemes every 6 months. So if you miss this particular scheme that's open now, the next one will be opening in about March of next year. As I said, it's quite rapid. We'll close this round at the end of October, and we fully expect to be able to make awards by the end of 2021. So within about 2 months. Just have a few interesting stories that have arisen from some of these schemes. We've had about 81 projects, quite a few SMEs, seems to be more every year that are accessing the scheme. And the kind of projects have supported have been everything from killer experiments, high throughput screens, or production to support clinical trials. Just to clarify a few things about the scheme, we don't take any IP in the project. We don't want any. This is not our job to be IP owners or managers. We are facilitators of access. The IP arrangements will very much depend on the facility the project is seeking to access. It's important to note that the vouchers are not intended to pay for a whole project, but they're meant to contribute and subsidize rather than fully fund a whole project. It's crucial that the applications must include a quote for services from the facility. So if you're looking to apply for a voucher, your first step is to contact the facility you'd like to access and then start that conversation. So in terms of amounts, we're now offering the voucher size in increments of $10,000 up to $50,000. But the applicants must match or nearly match, depending on the maths of the voucher value. Must match or nearly match the quote. So we're looking for approximate matching funding. Must be some skin in the game. So very briefly, the process, there are three parties to this process: the applicant, the provider, which is one of the facilities, the TIA. There is an online application that's open now. The applicant applies for the voucher with the agreement, written agreement in cooperation of the provider facility. This can be a PDF of an email saying, yes, I agree. This is a application we support. TIA assesses the applications, awards the voucher. Then the applicant and the provider enter into an agreement that takes that voucher value into account. The project starts, and it's at that point the applicant and provider let us know the project has started. Then TIA pays the facility, not the applicant. We pay the facility the value of the voucher, and then that saving is passed on. And we do this at the beginning of the project, not the end. At the end of the project, we ask for a brief 2-page final report when the work is complete, which we will keep confidential if that is the wish of the applicant. But it's a kind of thing we do like to shout about. So if it's a good news story, we do like to publicize that. Just finally, this is the page on which the current forms and guidelines and the link to the form currently at. And I encourage you to visit this page and have a bit of a look around. If there's anything that needs clarification, by all means, get in touch with us and we'll answer your questions. So I would just like to move on to the next part of this webinar, which is to introduce our guest speakers. First of all, I'd like to welcome Professor Loukas of James Cook University. He's going to be telling us a bit about his experience of accessing the Queensland node of the National Biologics Facility to assist his project. So delighted to welcome Alex. Is a NHMRC Senior Principal Research Fellow at JCU, alma mater of the University of Queensland, although he has worked at George Washington University and Queensland Institute of Medical Research, and also JCU, University of Edinburgh. His research, as you'll see, focuses on host-parasite interactions, using this knowledge to develop anti-helminth subunit vaccines and diagnostics, and also for biologics for treating inflammatory diseases. His work's been very well funded by Australian and US governments, philanthropists and venture capital groups, and he's a founder of Macrobiome Therapeutics, and I think he'll tell us a little bit about that as part of his talk. And he has received many awards for his research, including Fulbright scholarships at University of California, and is a fellow of Australian Society for Parasitology. And it's my pleasure to welcome Alex and ask you a little bit about your project and your experience of engaging with the scheme and the NBF. So thank you very much.
Thank you, Stuart. Can you see my screen and hear me okay?
Yes, on both.
Excellent. All right. So thanks for the introduction, Stuart, and Ben for introducing biologics and the AIBN node in Brisbane of the National Biologics Facility, which is the facility that we utilised for this project. So our program, as you'll see on the next slide, is a little unusual, and hence engaging the facility that we did was crucial, because what we're trying to bring forward into the drug development or discovery and development arena is something that that has not been dealt with before. And so we submitted a Pipeline Accelerator application, round one, through James Cook University in Cairns, where I'm based. And we requested 4 $5,000 vouchers to a total of $20,000, with the goal of expressing 3 of our lead proteins in mammalian cells at the NBF. And the goals of the application really were primarily to validate our own protein expression data that we generated in an academic lab, but using standards that resemble industry more than your typical academic laboratory at a university. Start at least to develop a purification process that's translatable into larger scale production, undertake an accelerated stability study, and provide my team here with the confidence that the proteins that we are working with have desirable properties for downstream development. You know, are they developable, or are there any red flags that we'd rather know now than many dollars and many hours down the track? And then, of course, generating a report that enabled a startup biotech that we founded, but essentially arming our investors with some external validation data that would allow them to continue to commit to investing. And so the goal of our project, just to sort of put it in perspective, is we work with parasites, parasitic helminths or worms in particular, which have been shown by many people over the last couple of decades to have potent anti-inflammatory properties. And we and others have shown that much of that anti-inflammatory prowess of the parasite, it resides within the proteins that they secrete into the tissues of their hosts, their human hosts, when they go about their business. And so the project was essentially, we had roughly 100 proteins that we'd identified by mass spec that the parasites secrete. We'd produced them in collaboration with Kirill Alexandrov's lab using a cell-free protein expression system. This was when he was at UQ, but he's now at QUT. And then we tested all of those cell-free products in a rapid mouse model of inflammatory bowel disease, of colitis, ranked our proteins for efficacy. And at this point of the project, we'd whittled it down to about 8 proteins, which we'd produced in our laboratory here at JCU in mammalian cells, in HEC293 cells. And then we took that further, showing human ex vivo data with biopsies from IBD patients. And it was at this point where we had our 3 leads, and we engaged the Pipeline Accelerator Program to really help us narrow this down to something where we can at least rank those top 3 and move into a formal preclinical development. And so our experience with the process of dealing with the NBF and through the TIA pipeline program was really straightforward. It was easy to prepare. The review process was rapid, and as Stuart's pointed out, it's every 6 months. The staff at both TIA and at the NBF were really responsive and excellent communicators, and they actually helped, as has been outlined earlier by Stuart, they helped us to craft the application, and so that it was, we really were able to design a fit for purpose study that gave us a good chance of getting the funding. And they also were really good in helping us tailor the studies after the award was made to get the most out of the possibilities. The data was also presented in a really comprehensive way that was easy for us to digest. And the data package actually did prove useful in convincing investors into our startup to get on board. And this is just a link to a website that we've formed for Macrobiome Therapeutics, which is now a startup biotech company that is trying to commercialize some of these helminth proteins for a variety of inflammatory diseases, including both inflammatory bowel disease, as I've shown you there, and we're now moving into the type 2 diabetes space as well. So thanks to TIA and to NBF, and I'll leave my presentation and happily take any questions whenever suits the audience.
Thanks very much for that, Alex. Yes, we'll see if there's any questions arise from the Q&A. We may have a bit of a discussion later on. So I'd just like to pass on directly to our final speaker, Mr. Greg Santamaria of Cyclotek. And if Greg is, here we go, fantastic. Greg, I just invite you to, I think you can just share your screen without any kind of...
Hope that works.
Yes, it does. Thank you very much. So just, so Greg's the, I think the part owner, director and CEO of Cyclotek Proprietary Limited and its subsidiaries, focusing on the manufacture of diagnostic radio pharmaceuticals for cancer and neurology. Cyclotek has grown to 6 facilities in Australia and New Zealand with about 80 staff and expanding into therapeutic radiopharmaceuticals for the Southeast Asian region as a whole. Greg's also well across the trials and tribulations of research infrastructure by being on the board of ANSTO, and is also a director of 302 Australia Foundation, to focus on the supply of fit-for-country oxygen systems, which is very relevant in the time of COVID, into sub-Saharan Africa and other regions. I'm delighted to welcome Greg to talk through an SME perspective of engaging with TIA facilities and the voucher system in particular. So take it away, Greg.
Thanks, Stuart, for the invitation to present to the community today. I appreciate that. Just a correction, not on the ANSTO board, one of the subsidiary ANSTO boards for ANSTO Nuclear Medicine Proprietary Limited. So that's what I was doing earlier this morning. But thank you anyway. So brief history of Cyclotek. There we go. As Stuart pointed out, 6 facilities across the Eastern Seaboard in Wellington, 8 cyclotrons. We're up to 85 staff now, and if my HR manager has her way over the next couple of days, it'll be 90. We manufacture typically midnight to 9 AM in our domain of manufacturing radiopharmaceuticals that have half-lives of 109 minutes and an expiry anywhere between 6 hours up to around about 12 hours would be the max. The main products we manufacture are the standard fluorodeoxyglucose, which is rebated by the federal government through the NBS system. Recently, we submitted an MSAC application with AANMS for rebate for prostate cancer. And so we're hoping for a favourable outcome for that submission shortly. And we manufacture 9 neurological tracers, 3 oncology tracers, and we have a tracer for activated T cells. So around about 13 products we manufacture routinely within a month at most of our facilities, but predominantly at our main facility in Melbourne. Some of the activities we're working on at the moment with Bristol Myers Squibb's cell therapies, and I noticed, Stuart, you had cell therapies listed on one of your projects there, but we developed with cell therapies and RMIT a process of taking a nanoparticle, copper-64, onto one of their CAR T cells. So an exciting project. We're actually doing some more work with them recently. We're moving into therapeutics, so a fair bit of research work in developing GMP grade lutetium PSMA and lutetium dotatate activities. And again, down the bottom there, we've got our engagement with MIPS, or the AAT-MCF, which is a mouthful, but I'll stick with MIPS if that's okay. So just quick understanding for those who may not understand, cyclotron develops our isotope which is predominantly fluorine across our companies. We go into hot cells, lead line hot cells with synthesis units in place and dispense through a grade A dispensing cell to achieve a GMP qualified end product. So what we were trying to do through our relationship with MIPS, and our relationship with MIPS goes back to 2000 and I think it was about 2005 when we had a CRC and we were involved with Monash back then. But from 2012, we sort of went quiet on research until more recently, our Chief Medical Officer, Dr. Rob Ware, has taken on a greater activity role in developing strategies around some novelty around bettering patient outcomes. So we approached MIPS on the basis of 3 identified commercial problems that we've got. We've got a new PSMA molecule, but unfortunately it's a 2 precursor, 2 reactor vessel. And in the commercial world, that's actually annoying for us. It may not be in the research world annoying, but we sort of like 1 precursor, 1 reactor vessels to make our life easier. So we've done significant work with MIPS in developing a single precursor onto a single reactor process. So difficult to ask, bearing in mind that MIPS have had 0 relationship in the fluorination or radioactive fluorination. But Jonathan and his team have got tremendous knowledge in the F18, F19 world. So that created an opportunity for us to see if they can do some cold work, which we could then replicate within some hot work within our confines of automation and synthesis yields. So that strategy has been well and truly evaluated. Mixed outcomes. So we haven't got it perfectly up to where our current 2 precursor, 2 reactor vessel sits from a production point of view, but early days for that development. Expansion of that work is not a priority right at this moment. We're going to take the current molecule into preclinical and first in human studies Q1, Q2 next year. The next one we had was a commercial opportunity. At the moment, there's a gallium dotatate for NETs, neuroendocrine tumors. We were looking for a fluorinated version so that we can better support our customers who don't have gallium generators. So that work has been successfully worked on, but GE and a few people overseas have beaten us to the mark, and we're now in discussions with them to see how best we can translate that activity over to Australia. The beauty about that development work was it greatly assisted our knowledge and capability within our 3rd project, which is the fluorinated radio labeling of a drug called enzalutamide. And this project has actually gathered tremendous pace with MIPS and the Cyclotek team, both working in hot and cold attributes. And what was fantastic was having MIPS researchers at Cyclotek and Cyclotek researchers at the MIPS facilities working collaborative. So that program has been very successful. We're now translating that to Cyclotek, where we're replicating, I think we've now replicated it 6 times. And now that's being worked on for a mouse model that Rob Ware is designing for later this year, and working on a biodistribution study of first in human in Q1 next year. So overall, the program has been very successful for us. The concept for us, this was a first go at working with MIPS on a one-on-one basis. They recommended the TIA program so that we would actually move forward. So this was sort of like a bit of a, hey, Greg, we can help you meet the costings of your 3 identified projects by using the TIA program. Bit skeptical at first. Grants are always hard. They seem to be always a lot of administrative work for some time, very little gain. What we found about this program was the ease of operation and the ease of completion. So I think that's 2 important things for commercial people. The IP, I think, Stuart, I saw that in one of your slides, was a huge advantage for Cyclotek to engage, because you'd appreciate we would like to control the IP. We found the MIPS leadership team to be very knowledgeable. They steered us, they helped us craft how best to design our projects and work across the paradigm of research and commercial chemists, which what we found, one of the interesting things we found is the research chemists weren't aware of some of the legal limits of excipients or components that are allowed within the final product. So whilst some of the researchers were using buffers and solutions and chemicals that may have solved the problem, but in a commercial world, they didn't actually meet the legal limits for some of those capabilities. So that was a good learning curve between our two disciplines. But overall, the TIA program, recommend it to any industry partner. It helps build the research program in the hope that we get better leverage. We were lucky that we had 2 postdoctorants working on our 3 projects. It was an excellent outcome for our company. And I thought I'd leave with a quote from 2 of the postdocs. It was this was presented to the staff at Cyclotek. And if I was looking at the things that my staff respond in an email, because I can send an email and get no response, but one of the things I did was I sent this response to our team. Out of 85 people, I think I got about 40 people responding with it was great working experience. It was great to be engaged with the researchers and learning how they do their discipline. So I think it was good to present what Kathy said and the outcomes of our staff. It's actually turned our company a little bit more proactive towards investing more heavily into some novelty in research and development programs that we've got. And Stuart, thank you for allowing us to communicate our story and a great program in the TIA platform.
Oh, thanks very much, Greg. And I really appreciate you taking the time, and Alex as well, to come and let us know how it went. It's really great to get that feedback and to know that what works and what doesn't work, and the fact that it's helped is terrific. So I think on that note, I'd like to open the floor up for any questions that anybody might have, either through the Q&A system, or even by perhaps raising your hand, and I might be able to find you and switch on your microphone. But if there are any particular questions on the scheme or any of the speakers that have gone, now's the chance. I did notice there was one from, just a note from Kayleen Simpson on the diagram of NCRIS capabilities. I think this is just to note, this is a concept that we've been working on recently. The fact that we already work together with other NCRIS projects in the omics space and the population health research space, we do that as a matter of course, but of late we find that it's of value to actually publicize and articulate that we do work together, and we can make referrals not just within TIA to the right facility, but also if a project seems like it needs some bioinformatics support, we'll put that onto bioplatforms. If there is an imaging aspect to a project, we'll put you in contact with the imaging facility or microscopy or anything like that. So just bear that in mind, that when you're talking to TIA, you're also talking to the rest of NCRIS.
Stuart, there is a question in the Q&A about kind of support available for developing drug-carrying nanoparticles and contrast imaging agents. Is that? That's something that
yes, thank you. This is from Chow Chow. Thank you for the question. I think on that specific technical questions, we've been in contact with you on other aspects. So we'll reach out to you on the specific on this technical question and have a discussion, and possibly put you in touch with some other NCRIS projects, particularly in the imaging agent area. I know that there have been some work, particularly in the medicinal chemistry facility, that is looking at imaging agents as well. So I think we'll take that offline if you don't mind, and reach out to you and discuss a little bit further. Have a question. Actually, I have a few questions on the base facility from Jeff Dunstade, asking how far down the development pipeline will the new RNA production facility go? Will it be making clinical material? And likewise from Leon Brownrigg over in Perth. Hey, Leon. On the type of nucleic acid to be manufactured by base. So if I can pass it on to Ben to address those questions.
Yeah, no, thanks for that. So it is still early days in terms of the facility is in the process of hiring the full complement of staff, and the capital plan to bring online the $1 million+ worth of equipment. Some of that, as you know, with global supply challenges we have right now, they're actually coming later in the year. But we were able to kickstart off the design, the sequencing, and the design elements of it. So what's happening right now, Jeff and Leon, is it's mRNA. So that's through in vitro, and we'll be looking into the sort of obviously small scale and kit-based purification, but then looking to make sure that that's scalable into sort of 15 mil IVT reactions, and that we can go through TFF filtration and chromatography based purification strategies, so that these are applicable for scale up, and we're not just dealing with sort of nucleic acid kit solvents and so forth. So yeah, the facility here where this will be produced is not going to be relevant for clinical material. So it will be, able to do up to the sort of milligram and I guess gram scale. The PEF facility has already, sort of made close to a gram of mRNA in the last calendar year, and double-stranded RNA. So they've been actively producing, also producing the enzymes to support this. So looking at other ways to try and bring down the cost structure. When you're using these substituted, specific, there's a lot of cost that goes into it. So in many ways, yeah, this is really about mRNA and double-stranded RNA capability. There is also an aspect of looking to also see about synthesis of DNA, so short form RNA or short form DNA and RNA, guide RNA and so forth. So, but again, some of this is dependent on the equipment that we bring in. And I guess finally, the formulation, it's not specifically, we haven't bought in, a formulation piece of kit. We have our eye on some, but it's based on some funding that'll come through, and it'll work with programs on a sort of an as-needs basis to try and see if the full component of how we make a packaged nucleic acid that could go into an animal trial or a clinical trial will partner with other people, and that's kind of what we're doing for this first program. But happy to talk about it later or offline. It's definitely a new and exciting space, and we're aware that this is going to be something that a lot of people are interested in, and we'll want to see what's available, and we welcome input and inquiries, because that's how we can better cater this capability. It's not going to fit for all sizes, but hopefully it will help move the needle for some companies that are looking to try and get into this mRNA and otherwise nucleic acid end.
And just to note that we also have this biologics survey that we have open at present, and that's a good way to engage with us and let us know where the particular gaps are at a big picture level. So although this is the base facility is a recent investment into space, it's not the last. but we do need some, I think, some consultation and some guidance from the community of potential users onto where the best strategic investment in this space are best taken as a next step. And this is all against the background of the current road mapping process is currently underway. So if there are any other questions, they'd like to put their hand up or add something into the Q&A. While people may or may not be tapping away, just to point out the closing date for this round of the accelerator scheme is going to be the 31st of October, the end of October, and the as Greg and Alex has noted, it really is a rapid turnaround. Our process of assessment is an internal, our three leads will examine and sort of advocate, if you like, for each of the applications, look for any missing information and follow up if necessary. Then we have an assessment panel of some of our independent board, which includes previous Chief Medical Officer of Australia and some external people as well in the pharmaceutical space, and somebody from MTP Connect to provide on the business potential business aspects to the application. We're able to do this very quickly, and as I know, we're looking to make awards by the end of the year, just in time for Christmas. Okay, so we're edging perilously towards 1:30. It's the end date, end time, I should say, for this event. And just to put in the chat window, there is a link to the biologics survey background and page. There's also a link to a post that describes the opening of the accelerator scheme and links to the guidelines and FAQ and the application form. Just notice that the application form this time around is completely online. So we'd advise you to use the template to develop an application and then simply cut and paste the plain text into the application form and then submit it, because you cannot save the form and come back to it. If you do wish to amend a form that you've submitted prior to the submission end date, or want to resubmit or amend it, please let us know. It's the first time we've used this online system, so there may be some tweaks to be made. Okay, so if there's no other questions, please feel free to reach out to us offline through the various means that we've described. It just remains me to thank everybody, all the speakers, our three capability leads, John, Ben, and Heather, and very special thanks to Greg and Alex for coming on and telling us their experiences with the scheme, and also to Stella and MJ for helping to develop the agenda and the speakers and keeping things on track and making sure this ran as smoothly as it possibly could, notwithstanding one or two inevitable technical glitches. But thanks very much to everybody, and I'd like to wish you the keep safe wherever you are and have a great rest of the day.
Thanks.